Hello and welcome everyone. This will be our final podcast on extensions to Mendel's rules. In this podcast we're going to talk about gene interactions, and more specifically we're going to talk about epistasis. So we're going to begin talking about some general features of gene interactions. Remember, when we first introduced Mendel's ideas, we talked about the dihybrid cross, where we had the round yellow seed, the heterozygote here, and we crossed it to the round yellow seed. And we got that classic 9 to 3 to 3 to 1. Now the reason we got this ratio was because of independent and we didn't mention this other part, was that each gene was in an independent pathway. If you had two genes involved in coat color, we might have seen slightly different results. We now know that because of gene interaction, and when we say gene interaction, we don't mean the actual genes themselves, the DNA is interacting. What we mean is that the proteins that are made from those genes interact with each other. And because of gene interaction, we know that one gene can affect a second gene. And the example we're going to talk about with this is epistasis. And we can define this as two or more genes that interact to control the same trait. We say that if one gene at one locus, so one gene, prevents the expression of a second gene, then the first gene this one here is epistatic to the second. Second gene. So when you have this pathway of genes that are interacting with each other, and if one of those genes is mutated and the other one is not expressed anymore, we say the first one is epistatic. And let's walk through an example. Alright, so let's walk through this example of Labrador coat color. For thousands of years, dog breeders have been selecting dogs for certain traits. And certainly, looking for dogs with certain coat colors has been something breeders have looked at. In 2004, the dog genome was sequenced, and this has really helped facilitate the way dog breeders do their work. But we can use it today as an example of epistasis. And we're going to talk about two genes, B and E. We know that B is dominant to little b. So a dog that is big B and either little b or big B is going to have a black coat color. A Labrador that is little b is going to be a brown lab, or a chocolate lab you might have heard of. How do we get a yellow lab? So let's draw out this pathway. There is a brown pigment that is produced, and this brown pigment is turned into a black pigment due to the expression of the B allele. So a dog that is either big B little b or big B will be black. It will have the black coat. A dog that is little b doesn't have this gene here, and so the pathway stops here. And so the dog would be brown. So little b is here. They lack the gene that makes a protein necessary to convert the brown pigment to the black pigment. So how do you get a yellow lab? Well, it turns out that there's this yellow pigment. Again, I don't have a yellow marker, and if I did, it wouldn't show up very well. And it turns out that this yellow pigment is converted into a brown pigment by the gene E. In this example, let's go ahead and write this up here. The gene E is epistatic to gene B. What that means is, if this gene is mutated, it doesn't matter what this gene is over here. So let's write down some possible genotypes then to help explain this. We know a black dog, as I say over here, is big B and either another big B or a little b. But it turns out in order for it to be big B, it also has to be big E. It could also be little e, doesn't matter what the other one is. To be brown, you have to be little b, but you also have to be big E. Because if you're not big E, if you're not changing this yellow pigment into brown, you're never going to make brown. To be yellow, you have to be little e. And it doesn't matter what you are at the big B. You could be big B, and you're going to be yellow, or the dog will be yellow. So big B, big B, big B, little b, little b, little b. If it's homozygous recessive at the E, it's going to be yellow, because E is epistatic to B. By mutating the function of E, it blocks the expression of B. Once you have this mutation, it doesn't matter what this gene is here. If it's big B or little b, doesn't matter. You're not making this intermediate. If you don't make the intermediate, there's no way it can continue down the pathway. E is epistatic to B. Alright, that's all I'm going to say about epistasis, but we will certainly talk about this in class, and there's a problem we'll definitely work on in class. Okay, well that ends this podcast on epistasis and gene interactions. And that ends all the podcasts on the extensions to Mendel's rules. As a quick review, as we look at this list, you should be familiar with the general definitions for all of these. You should be comfortable at looking at data and then determining if this extension is an example of lethal alleles, pleiotropy, phenocopy, sex influence, sex limited, environmental factors, gene interactions, discontinuous, continuous traits. You should be familiar with the differences between expressivity and penetrance as well. These are all extensions to what Mendel would have predicted as far as the phenotypes are concerned. Remember, his genotypic predictions hold true. There's nothing wrong with that. And that's really what his predictions were. We just have to sometimes interpret the phenotype slightly differently. Alright, that's all I have. If you have any questions, please make sure you come see me. I'll see you in class. Bye.